How are you guys? It's Friday yet. It is. I am having a Monday on a Tuesday. Oh my goodness gracious, you guys. I was supposed to bring Maisie, as you know, and then couldn't because I go out to my car and my car is broken. With a brick. Mikhail, you put that out into the universe and it came and got me. Not with a brick though. No, I go and it started up just fine and I go to shift it into reverse and nothing happened. Shifted into drive and nothing happened. It won't shift out of park. So had to quickly call an Uber and the Uber would not take a dog. So I could not bring my dog in the Uber. So that's why she's not out there right now. I know you were all worried. Oh my God, where's Miss Huff? Where's Miss Huff? Is Miss Huff okay? I know you were concerned. I'm fine. You guys didn't even notice. Kidding myself. So yeah, had to quickly get an Uber to work this morning.
You look far? Is that expensive?
It was about $25. Yeah, it was like 22, and then I had to pay the tolls because I add that onto your bill. So yeah, it's going to be about a $50 Uber day. And then I had to call and get my car towed to the mechanic place. So luckily my house is automated. So I have like my garage doors, I can open and close it from here. I have all my security cameras. I was able to watch and see what was going on. So that was at least good. So we'll see. I think it's probably the shifter cable. We don't know what that is. I'm assuming it's the thing that makes it go. So we'll see. So needless to say, my brain has been a little today. All right, so let's do a wee bit of review. We've been talking about passive and active transport, getting stuff in and out of our cells. Passive transport, remember, is going to be dependent on what we call the concentration gradient. That is the difference between the highest of the high and the lowest of the low. The steeper the gradient, the faster we move, just like our slide. The taller the slide, the faster we move. We had our diffusion, facilitated diffusion, osmosis. Now remember, osmosis is going to be the movement of water. It is unlike the others because the others are moving the molecules. Osmosis is moving the water. And then finally, filtration. These were our active transport. This is what does require energy. This is getting stuff into a cell. Endo, meaning in, cytosis, the cell. We had phagocytosis. Tell me what phago. What does the medical term phago mean? To eat. So we are taking in solid molecules. Pino. Is that our liquid? Yeah, it's hooked on phonics, guys. It's right there. Liquid. We had our Pinot Grigio. I'm going to be drinking a lot of that tonight, I think. And then we had our receptor mediated. This is lock and key. Usually, you can put your key in and have your cell work. Mine did not work this morning. But your receptor mediated, this is where we have very specific lock and key. Only specific things are allowed into the cell. We had our pictures. What goes in must come out. We don't just have toxins like cell poo and cell pee that has to be removed from the cell. We are also making products. We're making proteins. We're making things that the body needs. The cell will send those, remember, to our Golgi body or Golgi apparatus that will package them up into a little vehicle or a vesicle. And that vesicle, it would just do the opposite. When we're bringing stuff in, we bind with the lipid bilayer and bring it in, and the exact same thing is happening in the opposite direction. We have our transport vesicle, we bind it with the layer, and we are able to release the materials out to either be removed from your body or sent to somewhere that it needs to go. All right, so now we're going to talk about cell division. There are two different ways that cells can divide. and that cells can multiply. Meiosis, we're not going to concentrate too much on meiosis in this class. You guys will get into this a lot more when you get into some of your other classes. But meiosis is going to be where we have a contribution of DNA from two different cells that end up making a unique cell. You are a product of meiosis. You had a contribution from your mom, you had a contribution from your dad, bippity boppity boop, here you are. And this DNA, this unique DNA, will never again be replicated in quite the same way. Mitosis is the way that we are, or the way that cells divide that we're going to discuss in this class. The difference between mitosis and meiosis. Meiosis is a unique cell that is produced. Mitosis is a perfect, exact replicated copy. This is important that we do get an exact replication because we need the cell to act the same way. Let's take my liver. I'm going to take my liver out for a drive tonight. Not literally, obviously. Let's take your liver cells. Does anyone happen to know what your liver does? It does have some filtration capacity to help filter out some toxins. The other thing that the liver does is helps to convert nutrients into something that you can use. When you eat that Chipotle burrito and you're taking in the chicken and the sour cream, I don't know what you like on your burritos. Mine are pretty simple. I do the chicken, the sour cream, a little bit of lettuce, some cheese. That's about it. My burritos are very tiny compared to my significant other who gets everything and they can't even wrap it. It's all alien material though. My body doesn't know how to use any of that. It does break down the chicken into amino acids. It breaks down the sour cream into carbohydrates, et cetera, et cetera. That's not currency that my body recognizes. So if you were to travel to a foreign country and all you have is American money, what is the first thing that you need to do? I got to exchange my money. I have to convert my money. That's the bank of liver. The liver is going to take all of this foreign material and exchange it into something that your cells will accept as currency. Your cells are very greedy and they will do nothing for free. Everybody gets paid. And the currency that we are using to pay ourselves is glucose. And so your liver takes all of these little components of the nutrients and reassembles them into something that your body is able to use. So without your liver, nobody's getting paid. What do you think happens to cells? Well, let's see what happens to you. If you go to your job this evening, if you have a job, you go to your job tonight and they're like, hey, we're going to need you to work your shift, but we're not going to be able to pay you today. See ya. No. You're not going to want to work for no pay. Same thing with your cells. They'll stop working. So the liver, it's very important that the liver cells know how to do liver cell work. If you have a liver cell that replicates incorrectly, and now we have a liver cell that does not know how to do liver cell stuff, suddenly that unqualified liver cell, the non-working liver cell, now he's dividing. But he's dividing into liver cells that don't know how to do liver cell stuff. And they're dividing and they're dividing and they're dividing until eventually you have all these cells that are making up your liver that don't know how to do the work of the liver. And so that, bam, I've just described cancer. So it's really important that we have an exact genetic copy of the cell. This exact replication of the DNA is kind of the blueprint. It's the work manual. It is what tells the cell how to do its job, how to work, what to do. So mitosis, we need that exact replication of the cell. I love it. Tiana, your cat is killing me over here. Look, he knows we're talking about him. Look, that tail starts to flick. Loki! That tail is flicking angrily right now. What? Look at him. What? What do you want? Oh, I love it. All right, so let's talk about how these cells are replicating and becoming exact copies. So the first phase is called interphase. Well, not even really the first phase, because we're not doing any replication during this phase. Interphase is just where, let's keep talking about our livers. Interphase is where your liver is just walking around, doing his liver stuff. You know, he's got his little liver cell family, and he's doing a liver cell job, and he's just doing liver cell things. There is no active replication happening during interphase. And this is the stage that most of our cells spend most of their lives in, is that they're not actively dividing. It's not until they get a message from the body that says, hey, we need you to make some more of yourself, that then they will turn and start replicating. This, we used to call this the resting phase. I think we talked about this yesterday. This resting phase, kind of a misnomer, because cells are never truly at rest. Cells are either dividing or they are working. That's it. It's kind of one of those, they're either dividing or they're not dividing, and they're just doing their normal cell stuff. So we decided to go ahead and change the name. So instead of the resting phase, we call it the metabolic phase. Metabolic just means going about doing our daily business. During interphase, we do still have a nucleus, and all the genetic material is contained within that nucleus, and that's interphase. So what ends up happening as the cell transitions from interphase into kind of a pre-division phase? The first thing that we have to do when we want to divide our cell now is make an exact copy of the DNA. This is during the tail end of this interphase, entering into the division phase. Here we have our mother cell, and we're going to make an exact copy, an exact replication. We're going to talk about how this happens. And then we can divide, and now each of the cells, the parent, and she's called the daughter, I don't know why they don't call it the son, it's the daughter cell, they each have now an exact replication of the DNA. And now either one of these can start dividing if they come out of interphase. So all of this DNA replication, gearing up to actively divide, is going to happen during this interphase. Couple of sub-phases that we're going to talk about. That's a lot of words. Okay, don't panic. We're going to break this down. We're going to simplify this. We have, I'm going to take a load off for a minute. That feels good. I got DNA. It's a terrible DNA strand, but you get the idea. We've got this double helix. And what we need to do, we have our nucleotides, right? They're matched up. Apple tree, put your car in the garage. Although if you do that, it may never come out again. So
You're really hurt over that.
I am. It was just, you know how you have a routine in the morning? And when something goes out of that routine, you just feel off for the rest of the day. And yeah, that's just where I am. I feel off. I forgot my keys. So I usually have my lanyard and I have the weight of my keys. So I couldn't even get into the side of the building. My office is locked. Don't have my keys. I'm discombobulated. That's a great word. I'm discombobulated right now. All right, so I have my nucleotides. Apple tree, put your car in the garage. But what I need to do is I need to separate these strands. Because do you see if I take these and I separate them now, okay, I unwind them. I have my nucleotides that are still matched up. I just unwound it. Now I can come through and I can buzz it right down the middle. If I have half of this DNA, can I make an exact copy out of just that bit of information? Sure I can. And if I take this half and donate that to the other cell, I can also make an exact copy. And so this whole slide is just describing how does this happen. Well, do this. Here's my unzipped DNA. Here's my nucleotides. The first thing I gotta do is I have to unbond these nucleotides. I gotta undo them. So I have this thing called a helicase. This is an enzyme. Remember, anything that ends in ACE, generally an enzyme. And basically what he does is he comes through and he is a kind of a bulldozer, and he breaks all of these bonds and he separates this into two distinct strands of DNA that we can now reassemble. The way that we reassemble is kind of interesting. I find this really fascinating. So here's my, you know what, I'm gonna do this. This will be much easier to see. All right, so here is my terrible DNA, and I'm unzipping it. I have my helicase. Here's my helicase, bulldozing and unzipping here. Well, the way that I'm going to reconstruct these two strands is very slightly different, and it has to do with a sugar. Sugars, kind of like this, they're 5-pointed, and the way that they are assembled in this DNA determines how do I reassemble the DNA. This strand here, I can just reassemble this way, reassemble, no worries at all. This strand here, I have to reassemble in the opposite direction. And so what that means is I reassemble, and then I have to come here, jump, reassemble, come here, jump. So instead of nice and smooth, I am doing it this way, reassemble, jump to here, reassemble, jump to here, reassemble. It's a very jerky move. It's not very smooth at all. This nice smooth in the correct direction is called the lead strand. This one, now that we are all computer experts, you ever heard the term lag? When your computer is laggy and your video is kind of stuttering, it's like a buffer. That's what this one's called, the lag strand, the lead strand and the lag strand. I do have a little fun Amoeba Sister video to show you about how all this works. Erase all of that. So that's all this is saying, is that we have the helicase that is unzipping it, and then we have the reassembly of this DNA. One of them is being done continuously, the other one is being buffered because of the way that the sugars appear in the DNA. All right, before I do this, I want to show you guys this little this. All right, let me show you this cute little Amoeba Sister video. Oops. Make sure that I'm sharing my computer. All right. Oops, that's not the one I want. Sorry. All right, you in TV land, can you guys see the video? All right, and then who am I looking at? Tiana, I'm still looking at you. Give me a thumbs up if you can hear it.
DNA. We talk about it so much, it's the ultimate director for cells, and it codes for your traits. With a molecule that has a function like that, it makes sense that when you make another cell, like in cell division, you would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where? Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when? When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, that's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great video game. Still waiting for that to be invented. I'm going to introduce the key players in DNA replication so that you can get some background information. Now remember, these are just some major key players. There's a lot to this process. Many of the key players are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it's an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So here we go with the key players. Helicase, the unzipping enzyme. If you recall that DNA has two strands, you could think of helicase unzipping the two strands of DNA. Helicase doesn't have a hard time doing that. When unzipping, it breaks through the hydrogen bonds that hold the DNA bases together. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer it makes? The primer is actually made of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that later on. Now, don't feel overwhelmed. We'll go through the basics of this sequence in order. But remember, like all of our videos, we tend to give the big picture. There are always more details and exceptions to every biological process that we can't include in such a short video. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. At the origin, helicase, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, bind to the DNA strands to keep them separated. And topoisomerase, I always have to slow down when I say that enzyme's name, keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next steps. Primase comes in and makes RNA primers on both strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They're not identical, they complement each other. In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes with the base thymine, and the base guanine goes with the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3' or 3' to 5'. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of this ring structure. Now, you do the same thing for the other side, but keep in mind DNA strands are anti-parallel to each other. So let's count these, again clockwise after the oxygen. 1', 2', 3', 4', 5'. and the 5' is out of this ring. This strand on the left runs 5' to 3', and the strand here on the right runs 3' to 5'. We'll explain why all that matters in a moment. So let's take that knowledge there and look at DNA replication here. In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strands. Now, the thing about DNA polymerase is, when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase once again is building a new strand in the 5' to 3' direction. But there's a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take care of the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the two copies each contain one old original strand and one newly made one. One last thing. Surely you've had to proofread your work before to catch errors? In this process, you don't want DNA polymerase to make errors. If it matches the wrong DNA bases, then you could have an incorrectly coded gene, which could ultimately end up in an incorrect protein, or no protein. DNA polymerase is awesome. It has proofreading ability, meaning it rarely makes a mistake, which is a good thing. So remember how we said there is far more detail to this process to explore? The detailed understanding of DNA replication has led to some life-saving medical treatments that can stop DNA replication in harmful cells, including pathogenic bacteria or human cancer cells. We encourage you to explore beyond the basics. Check out the further reading suggestions and the video details to explore more. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, did that kind of explain the whole leading strand and lagging strand? I have one more kind of interesting video. I think you guys have been introduced to this guy before. It's a crash course. I think Ms. Rivera or maybe Ms. Cat. I love him. I think he's amazing. What am I doing? All right, so he's done. Now this is just kind of the exact same information,
It's just beautiful, isn't it?
Little bit more in depth, but I really love the way that he explains this as well, and it really kind of solidifies that leading and lagging strand concept.
It's mesmerizing. It's double helix exciting. You really can tell just by looking at it how sort of important and amazing it is. It's pretty much the most complicated molecule that exists and potentially the most important one. It's so complex that we didn't even know for sure what it looked like until about 60 years ago. And so multifariously awesome that if you took all of it from just one of our cells and untangled it, it would be taller than me. Now consider that there are probably 50 trillion cells in my body right now. Laid end to end, the DNA in those cells would stretch to the sun not once, but 600 times. Mind blown yet? Hey, you want to make one? Of course, you know, I'm talking about deoxyribonucleic acid, known to its friends as DNA. DNA is what stores our genetic instructions, the information that programs all of our cells' activities. It's a 6 billion letter code that provides the assembly instructions for everything that you are. And it does the same thing for pretty much every other living thing. I'm gonna go out on a limb here and assume that you are a human, in which case every body cell that you have, or somatic cell, in you has 46 chromosomes, each containing one big DNA molecule. These chromosomes are packed together tightly with proteins in the nucleus of the cell. DNA is nucleic acid, and so is its cousin, which we'll also be talking about, ribonucleic acid, or RNA. Now, if you can make your mind do this, remember all the way back to episode 3, where we talked about all of the important biological molecules, carbohydrates, lipids, and proteins. That ring a bell? Well, nucleic acids are the fourth major group of biological molecules, and for my money, they have the most complicated job of all. Structurally, they're polymers, which means that each one is made up of many small repeating molecular units. In DNA, these small units are called nucleotides. Link them together and you have yourself a polynucleotide. Now, before we actually put these tiny parts together to build a DNA molecule like some microscopic piece of IKEA furniture, let's first take a look at what makes up each nucleotide. We're going to need 3 things. 1, a 5-carbon sugar molecule, 2, a phosphate group, and 3, one of 4 nitrogen bases. DNA gets the first part of its name from our first ingredient, the sugar molecule, which is called deoxyribose. But all the really significant stuff, the genetic coding that makes you, is found among the 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. It's important to note that in living organisms, DNA doesn't exist as a single polynucleotide molecule, but rather a pair of molecules that are held tightly together. They're like an intertwined microscopic double spiral staircase, basically just a ladder, but twisted, the famous double helix. And like any good structure, we have to have a main support. In DNA, the sugars and phosphates bond together to form twin backbones. These sugar-phosphate bonds run down each side of the helix, but chemically in opposite directions. In other words, if you look at each of the sugar-phosphate backbones, you'll see that one appears to be upside down in relation to the other. One strand begins at the top of the first phosphate, connected to the sugar molecule's 5th carbon, and then ending where the next phosphate would go, with a free end at the sugar's 3rd carbon. This creates a pattern called 5' and 3'. I've always thought that the deoxyribose with an arrow, with the oxygen as a point. It always points from 3' to 5'. Now on the other strand, it's exactly the opposite. It begins up top with a free end at the sugar's 3rd carbon, and then the phosphates connect to the sugar's 5th carbons all the way down. And it ends at the bottom with the phosphate, and you've probably figured this out already, but this is called the 3' to 5' direction. Now it is time to make ourselves one of these famous double helices. These 2 long chains are linked together by the nitrogenous bases via relatively weak hydrogen bonds. But they can't be just any pair of nitrogenous bases. Thankfully, when it comes to figuring out what part goes where, all you have to do is remember that if one nucleotide has an adenine base, only thymine can be its counterpart. Likewise, guanine can only bond with cytosine. These bonded nitrogenous bases are called base pairs. The GC pairing has 3 hydrogen bonds, making it slightly stronger than the AT base pair, which only has 2. It's the order of these 4 nucleobases, or the base sequence, that allows your DNA to create you. So AGGTCCATG means something completely different as a base sequence than, say, TTCAGTCG. Human chromosome 1, the largest of all of our chromosomes, contains a single molecule of DNA with 247 million base pairs. If you printed all of the letters of chromosome 1 into a book, it would be about 200,000 pages long. And each of your somatic cells has 46 DNA molecules tightly packed into its nucleus. That's 1 for each of your chromosomes. Put all 46 molecules together and we're talking about roughly 6 billion base pairs in every cell. This is the longest book that I've ever read. It's about 1,000 pages long. If we were to fill it with our DNA sequence, we'd need about 10,000 of them to fit our entire genome. Pop quiz. Let's test your skills using a very short strand of DNA. I'll give you one base sequence, you give me the base sequence that appears on the other strand. Okay, here goes. So we got a 5' AGGTCCG to 3' and...
All right, so how would we reassemble this? This is 1/2 of the DNA. How would we reassemble this and give, oops, I'll do it. I can't do it on there. And give our full DNA sequence. It goes here. There you go. Nice. Hold on. Easy enough.
And time's up. The answer is 3' TCC AGGC 5'. See how that works? It's not super complicated. Since each nitrogenous base only has one counterpart, you can use one base sequence to predict what its matching sequence is going to look like. So, could I make the same base sequence with a strand of that other nucleic acid, RNA? No, you could not. RNA is certainly similar to its cousin DNA. It has a sugar phosphate backbone with nucleotide bases attached to it, but there are 3 major differences. 1. RNA is a single-stranded molecule, no double helix here. 2. The sugar in RNA is ribose, which has one more oxygen atom than deoxyribose, hence the whole starting with an R instead of a D thing. And finally, RNA does not contain thymine. Its 4th nucleotide is the base uracil, so it bonds with adenine instead. RNA is super important to the production of our proteins, and you'll see later that it has a crucial role in the replication of DNA. But first, biographies! Yes, plural this week, because when you start talking about something as multitudinously awesome and elegant as DNA, you have to wonder just who figured all this stuff out and how big was their brain? Well, unsurprisingly, it actually took a lot of different brains in a lot of different countries and nearly 100 years of thinking to do it. The names you usually hear when someone asks who discovered DNA are James Watson and Francis Crick, but that's bunk. They did not discover DNA, nor did they discover that DNA contained genetic information. DNA itself was discovered in 1869 by a Swiss biologist named Friedrich Miescher. His deal was studying white blood cells, and he got those white blood cells in the most horrible way you could possibly imagine from collecting used bandages from a nearby hospital. God. For science, he did it. He bathed the cells in warm alcohol to remove the lipids, and then he set enzymes loose on them to digest the proteins. And what was left after all of that was this snotty gray stuff that he knew must be some new kind of biological substance. He called it nuclein, what was later to become known as nucleic acid. But Miescher didn't know what its role was or what it looked like. One of the scientists who helped figure that out was Rosalind Franklin, a young biophysicist in London nearly 100 years later. Using a technique called X-ray diffraction, Franklin may have been the first to confirm the helical structure of DNA. She also figured out that the sugar phosphate backbone existed on the outside of the structure. So why is Rosalind Franklin not exactly a household name? Well, 2 reasons. 1, unlike Watson and Crick, Franklin was happy to share data with her rivals. It was Franklin who informed Watson and Crick that an earlier theory of a triple helix structure was not possible. And in doing so, she indicated that DNA may indeed be a double helix. Later, her images confirming the helical structure of DNA were shown to Watson without her knowledge. Her work was eventually published in Nature, but not until after 2 papers by Watson and Crick had already appeared, in which the duo only hinted at her contribution. Even worse than that, the Nobel Prize Committee couldn't even consider her for the prize that they awarded in 1962 because of how dead she was. The really tragic thing is that it's totally possible that her scientific work may have led to her early death of ovarian cancer at the age of 37. At the time, the X-ray diffraction technology that she was using to photograph DNA required dangerous amounts of radiation exposure, and Franklin rarely took precautions to protect herself. Nobel Prizes cannot be awarded posthumously. Many believe that she would have shared Watson and Crick's medal if she'd been alive to receive it. Now that we know the basics of DNA structure, we need to understand how it copies itself, because cells are constantly dividing, and that requires a complete copy of all that DNA information. It turns out that our cells are extremely good at this. Our cells can create the equivalent of 10,000 copies of this book in just a few hours. That, my friends, is called replication. Every cell in your body has a copy of the same DNA. It started from an original copy and will copy itself trillions of times over the course of a lifetime, each time using half of the original DNA strand as a template to build a new molecule. So, how is a teenage boy like the enzyme helicase? They both want to unzip your genes. Helicase is marvelous, unwinding the double helix at breakneck speed, slicing open those loose hydrogen bonds between the base pairs. The point where the splitting starts is known as the replication fork. It has a top strand called the leading strand, or the good guy strand as I call it, another bottom strand called the lagging strand, which I like to call the scumbag strand, because it is a pain in the butt to deal with. These unwound sections can now be used as templates to create 2 complementary DNA strands, but remember, the 2 strands go in opposite directions in terms of their chemical structure, which means that making a new DNA strand for the leading strand is going to be much, much easier than for the lagging strand. For the leading, good guy strand, an enzyme called DNA polymerase just adds matching nucleotides onto the main stem all the way down the molecule. But before it could do that, it needs a selection of nucleotides that fill in the section that's just been unzipped. To get started at the very beginning of the DNA molecule, DNA polymerase needs a bit of a primer, just a little thing for it to hook onto so that it can start building the new DNA chain. And for that little primer, we can thank the enzyme RNA primase. The leading strand only needs this RNA primer once at the very beginning, then DNA polymerase is all, I got this, and it just follows the unzipping, adding new nucleotides to the chain continuously all the way down the molecule. Copying the lagging or scumbag strand is, well, he's a freaking scumbag. This is because DNA polymerase can only copy strands in the 5' 3' direction, and the lagging strand is 3' 5'. So DNA polymerase can only add new nucleotides to the 3' end of a primer, so maybe the real scumbag here is the DNA polymerase. Since the lagging strand runs in the opposite direction, it has to be copied in a series of segments. And here that awesome little enzyme, RNA primase, does its thing again, laying down an occasional short little RNA primer that gives the DNA polymerase a starting point to then work backwards along the strand. This is done in a ton of individual segments, each 1,000 to 2,000 base pairs long, each starting with an RNA primer. These are called Okazaki fragments, after the couple of married scientists who discovered this step in the process in the 1960s. And thank goodness they were married so that we could just call them Okazaki fragments instead of Okazaki-someone's-someone fragments. These allow the strands to be synthesized in short bursts, and then another kind of DNA polymerase has to go back over and replace all of those RNA primers, and then the little fragments gets joined up by a final enzyme called DNA ligase. And that is why I say that the lagging strand is such a scumbag. DNA replication gets it wrong about 1 in every 10 billion nucleotides. But don't think your body doesn't have an app for that. It turns out that DNA polymerases can also proofread, in a sense removing nucleotides from the end of a strand whenever they discover a mismatched base. Because the last thing we want is an A when it would have been a G. Considering how tightly packed DNA is into each one of our cells, it's honestly amazing that more mistakes don't happen. Remember, we're talking about millions of miles worth of this stuff inside us, and this, my friends, is why scientists are not exaggerating when they call DNA the most celebrated molecule of all time. So you might as well look this episode over a couple of times and appreciate it for yourself. And in the meantime, gear up for next week when we're going to talk about how those 6 feet of kick-ass actually make you you. Thank you to all the people here at Crash Course who helped make this episode
Like that one because
awesome. You can click on any of these things.
Oops. The animation that he shows for the lead versus lagging strand, I really like the way he went over that. All right. Let's get you guys back. Okay, so now that we have successfully replicated our DNA, we've successfully had our lead strand, our lag strand, everything has been reassembled. Now we get to start division. All of that previous happened during interphase. The preparation to divide is also happening during interphase. We have some preparatory things that we got to do. You can't just randomly divide a cell. There's stuff we got to take care of. So the first thing we got to take care of, okay, so think about this for a minute. I have a cell this big, and I want to make this into 2 cells this big. Don't I need to make this first cell bigger to do that? So if I want to make this into 2 that are the same size, if I want to make this into 2, don't I have to pretty much double this one's size in order to accomplish that? Otherwise, aren't I going to have 2 cells that are half the size? Of course I am. So the first thing that I have to do is I need the cell to get bigger. I need it to be able to split into 2 equal original sizes. And this cell growth is called the growth phase, the G1 phase. The cell will double in size and the organelles will also double. We gotta prepare. This is all about preparation. I'm making a new cell, which means not only do I need an exact copy of my DNA, I need things like endoplasmic reticulum and my Golgi apparatus and my mitochondria. I need all of the things to set this cell up for success. Once I have all of that taken care of, now I'm going to go ahead and actively do the lead lagging DNA replication. This is known as synthetic or synthesis to create. Once that's happened, my growth 2 phase, this is where I need to pack a lunch. Cell division takes an awful lot of energy, and cell division takes an awful lot of things like proteins, enzymes, and so this is where I kind of in my mind think, okay, my little cell is ready. He's ready to start dividing. He's packing himself a lunch, and he's packing himself a toolbox to do it. So growth 1, the cell is doubling in size and getting all the organelles ready and organized. Synthesis, we've gone ahead and replicated our DNA. G2, or the growth 2 phase, I got my toolbox together. I got everything. All the T's are crossed, all the I's are dotted, ready to go ahead and start active division. Oh, I have about 100 pictures of this leading and lagging. I don't know why I got so excited with all these pictures. The picture. So one of these pictures will work for you. Picture. Guess what comes next? Picture. Picture. So this is a chromosome. This is what we are turning our replicated DNA into. And essentially what this chromosome has is 2 compressed exact copies. So if I look here, what I'm going to end up doing is splitting this guy in half. This half will go to one cell, this half will go to the other cell, and that's how we're going to perfectly replicate this DNA. All right, so we've now exited out of interphase. Okay, we are prepped, we are ready, the DNA is hot off the press, our toolbox is packed, our lunch is packed, we've got the energy, we've doubled in size, we are ready to divide. This is the exciting, active part. As we are dividing, we do have some specific phases that we're going to go through. And these phases just simply describe a specific thing that is happening, a specific step in the division process. We have prophase, which pro meaning before, prophase is the phase that happens right before replicate or right before division. Metaphase, and then anaphase and telophase. So I'm going to go through each of these steps, and I'm going to describe for you exactly what is happening during this active division. Do I have any comments, questions, concerns so far? Is everybody on the bus? Fabulous. Fantastic. So the first phase is the prophase. So let's think about this critically. I have a cell, it's doubled in size. I have 2 strands of perfect DNA. I've got all of my organelles and I'm ready. The first thing that I need to do is get the DNA out of the nucleus, because the DNA is still in there. It's still locked away. So the first thing I got to do is release the DNA. And so what is happening during this prophase is just that. My DNA, now even my copy of that DNA, is trapped in this nucleus. So what do I need to do? Right? I need to get rid of the nucleus. I need to break down the walls and set the DNA free. The other thing that I have going on are these 2 little organelles that start spitting out these little fibers, these little tug-of-war fibers. So they start making these little fibers. Those are going to be important because that's what we're going to use to pull apart these 2 DNA strands. So during prophase, we're gonna see the nucleus start to disappear. During metaphase, the nucleus has disappeared. Our DNA is floating out free. Everything looks great. And what happens is these chromosomes very kindly line up right in the middle of the cell. Metaphase, middle. They line up right on these little tug-of-war guys, these little tug-of-war strands. This picture here, I love these pictures. I am so happy I found these pictures. This is a real cell going through the same phase. So here you can see, here's the cell wall. We don't have a nucleus, but we do have the chromosomes very clearly lined up. And if you look super carefully, you can even see these lighter purple. Those are the strands that are going to end up pulling them apart. It's an amazing picture. I like to sit and look at it. So cool. After the metaphase, we transition into the next, which is the anaphase. Tug-of-war time, tug-of-war time. These fibers start pulling apart our chromosomes, pulling one half to one side and one half to the other. Now remember, each of these halves contains a full DNA. And so this anaphase is tug-of-war time, pulling these two chromosomes apart and getting ready to place them into two individual cells. So our spindle fiber separates, pulling them apart. This is actually a really great picture, although sadly I had to make it smaller. But you can see here how we have here this dark purple right here, dark purple right here. Those are the two halves of the chromosome that is being pulled apart. Finally, we have the final stage of our mitosis, which is telophase. This is where my nucleus reforms and my cell has formed this little cleft, and my cell membrane is now going to reform and split it. You can see here, split now and divide, and we have two perfectly replicated cells. And then it happens all over again. Second verse. Really? Nobody? Second verse. Same as the first. Really? You guys are awful. So both cells, especially the new daughter cell, is going to immediately enter the metabolic phase and just start working like a liver cell, working like a nerve cell, or working like whatever cell she made. And then when the body tells her, she will go through this exact same thing. She will enter into the end of interphase, where she herself will enter G1. She'll get doubled in size, replicate her DNA, pack all the proteins and the tools that she needs. She will then enter into mitosis, prophase, where the nucleus disappears and the DNA is set free. The little fibers begin to form. You have metaphase, where the chromosomes are now completely free. We've got our tug-of-war fibers. The chromosomes line up in the middle. Then we have anaphase, tug-of-war time, tug-of-war time. And these fibers start pulling the chromosomes apart to opposite ends of the cell. And finally, we have telophase, where we rebuild the phospholipid bilayer, separating them into two cells, the mother and the daughter. Any questions about any of that? Are you guys really okay, or are you just like, shut up and stop talking, we want to have lunch? Not that you would say that to my face, but. Picture. This is just showing the stages. Here's our prophase. Nuclear envelope has disappeared. Metaphase, they've lined up. Anaphase, tug of war time. And here we have rebuilt the lipid bilayer, separating it into two. All right, I have kind of a cool, I think I do. Yes. Here's an animation just to demonstrate everything we just talked about.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.
And that's the story of mitosis. All right. How is everybody? Excellent. So we have some things that help to control this whole process. We don't want cells just randomly dividing when they don't have to. We only want them to divide when we need them to. And we have something called contact inhibition. So let me give you an example. Let's say this room needs to be filled with cells. And so every one of us is a little cell and we all start replicating until bloop, 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 bloop. This entire room we are standing shoulder to shoulder, back to front with each other. And it is this contact that tells us to signal, stop reproducing, we have enough. And so when these cells come in contact with each other, with the surrounding cells, this was their signal to stop dividing. Because otherwise, if we just keep dividing and dividing and dividing uncontrolled, we're going to slop out, we're going to pop out of the windows, we're going to pop out of this door, bam. That's kind of the definition of a tumor. Uncontrolled cellular growth. So that's how we control our cell division. That's it. Don't want anyone falling out of the window. Ouch. All right, here's what I want to do. I want to ruminate a little bit over what we've talked about today. We're going to get into this protein synthesis, genetic mutations, and finish this up tomorrow. I think we've only got a few slides left, if I'm not mistaken. Oh yeah, we've only got a few slides yet left. So I'm going to give you guys, here's what I'm going to have you guys do for the last, 10 minutes of class. I'm going to give you a little extra points, easy points, little in-class assignment. And I'll create a Moodle for you to hand this in. I want you guys to just take a blank piece of paper, and I want you to take your colored pencils, share if you don't have any, take your highlighters, markers, and I want you to draw and briefly label what's happening in each phase of mitosis, starting with prophase. So you're going to draw a little picture of prophase, metaphase, anaphase, and telophase. And you're going to color it in, and you're going to just do some keywords or make something on the paper that will help you specifically remember what each phase is doing. And then yes, you can use your notes. This is not a test. You can use your notes. This is just creating new pathways. The best way to do this is if you want to do it as a little story, if you want to do it as just a picture, whatever's going to help you remember these phases of active mitosis. I am going to put I'm going to go ahead and put an assignment on Moodle right now. I'm going to call it mitosis drawing. You guys are CD, right? Okay. All right, I have created an assignment in Moodle for you to hand that in right here, mitosis drawing. And I will give you the remainder of the class period, although I have set it so that you don't have to hand it in until tomorrow morning. So if you want to work on it a little bit, I don't want it to be rushed. I want you to really be thoughtful of your drawings. And I will give you guys the remainder of the class time. right, so you guys in TV land, you guys can use the remainder of this class time to do the drawings. And then you can either label or you can simply use colors, some sort of depiction that outlines what is happening during each phase, so that you'll have something in your mind when we start testing you over this later this week. You can hand this in on the Moodle, and otherwise I will see you guys here tomorrow. And I will see you guys in TV land. All right, so you guys have a wonderful day. Oops, have a wonderful day. And I will see you here tomorrow.